Now, Gregor Mendel is known as the father of modern genetics. He is the first individual to have come up with a way of determining the mathematical reasons for why traits are passed on to offspring. He was an Austrian monk, and a lot of what he discovered was just by own experimentation. It's not as if he was a person that studied science or anything like that, but these are some things that he had. He enjoyed studying plants, so that makes him a botanist in some respects, even though he might not have a degree in botany, but he was a monk at a monastery. They had a garden, and these are just some of the observations that he was able to make. Now, it's very fortunate that he studied pea plants, because had he studied anything else like higher organisms like ourselves or animals or things like that, then what we know about genetics probably wouldn't be as advanced as it is at this point. So anytime we're studying DNA and we are looking at how things are passed on to offspring, that whole science or branch of biology is called genetics, because the passing on of those items is heredity. Okay, we've just talked about Mendel and his characteristics, known as the father of genetics, Austrian monk. He worked with the garden pea, and again, a lot of what he discovered was being fortunate that these organisms were in the garden that he resided in. There are many different reasons why pea plants were beneficial. I told you a couple when we first started the lecture, but they have very simple traits, 7 main characteristics. They grow very quickly. They reproduce very often. You know, insects and plants can reproduce very often. Again, higher animals like ourselves take months to years to develop. So, you know, the pea plant was a very good thing for Mendel to study, and it's why we are where we are in our knowledge of genetics. Okay, Mendel took the traits that we just saw, the 7 characteristics. He took their parents, okay? They were pure for the particular traits that he was studying. For example, if he was looking at color, the flower color of the pea plant, then he was taking the parent that was purple and the parent that was white, okay? Neither one of them had a mixture of characteristics. They were purebred for their particular thing. So he took the pollen also, which is the male sex cell for plants, and he placed it into the female, or where it would combine with the female sex cells for plants. And that's called cross-pollination. When plants are able to pollinate on their own, we call that self-pollination. But when a scientist or a person like Mendel studying it actually manipulates it, it would be cross-pollination. Okay, in this slide we see the different parts for male and female. We also see a brush by which a scientist can do this cross-pollination, taking the pollen from one thing and transferring it to the female area where they're going to hopefully meet the sperm cell and the egg cell and produce another organism. Now, normally what happens is that insects or wind would do this kind of thing, okay? So when an insect like a bumblebee gets nectar from a flower, some of the pollen would brush off on the body, and then as it goes to other flowers, the pollen falls off, and that's how a lot of fertilization occurs in nature. So, we designate the letter P. Usually we don't use it in genetic studies, mostly because the letter P is designated for only the parental generation. And F1 is also called filial, so it's the first filial group, or F1. F2 is the second filial group, okay, or F2 as it is. Now, hybrids have a combination of 2 traits from both parents. And so sometimes you can kind of figure out that a heterozygous combination would also exhibit hybrid characteristics as well. All right, so what I want you to do is, yesterday when we were taking notes on the podcast, it was explained to you which letters we use, when, why we use those letters. So Round is the dominant characteristic. So we are going to use capital R to represent our dominant characteristic. So above round here, I want you to put 2 capital R's.
2 capital?
Above wrinkled, I want you to put 2 lowercase r's to represent the recessive characteristic. We are not changing the letter case, or excuse me, we're not changing the letter type. We're going to keep the same letter. We're just going to use the lowercase letter to represent the recessive. Here in this space, I want you to put capital R, lowercase r. Capital R, lowercase r right here next to F1. Right here, I want you to put capital R, lowercase r as well to represent the F1 parents that we're going to cross. Now, when we cross the 2 F1 parents, the offspring would be F2. And whenever you have 2 heterozygous parents, meaning a big letter and a little letter, whenever you combine those parents to try to get the F2 generation, you're always going to get this 3-to-1 ratio. So that 3-to-1 ratio represents the phenotype. Write that down there somewhere. The phenotype ratio. 3-to-1 here represents the phenotype ratio. So 75% possibility or probability that these individuals will be round. Then there's a 25% probability or possibility that it would be wrinkled. I can also say 3/4 here and 1/4 there. That is always going to be the case whenever we have heterozygous parents. Okay, here are Mendel's 7 traits again. And let me explain to you what's going on here in this. And this is a similar one to your book, so you don't necessarily have to take a picture of it. But we have the parental generations on the top. They are pure for their particular characteristic. Round is dominant over wrinkled. For seed color, yellow is dominant over green. Seed coat color, gray over white. For pod shape, smooth over constricted. Pod color, green over yellow. Flower position, axia over terminal. And for height, tall over short. Now, this is the result. These are the F1s. So these would be all big letter, little letter. These would be a combination of the top ones being all big letters and the bottom ones being all little letters. But that would produce the F1, which would be heterozygous. All right, so above the parent column, here's what I want you to write. I want you to write homozygous dominant. Then draw a multiplication or a cross mark, okay? So times homozygous dominant times homozygous recessive. Write those words above the column here. Homozygous dominant times homozygous recessive, because that's what, it didn't show up here on this slide for whatever reason, but actually on another diagram this should be long stems times short stems. Red flowers times white flowers. Stop talking. Green pods times yellow pods. Round seeds times wrinkled seeds. Yellow seeds times green seeds. All right, here in this middle column, I want you to put heterozygous dominant above the column there. Heterozygous dominant goes right here. Then above this third column, I want you to put 3-to-1 phenotype ratio. 3-to-1 phenotype ratio, heterozygous dominant, homozygous times homozygous recessive. Homozygous dominant times homozygous recessive. 3-to-1 phenotype ratio on the last one. All right, so principle of dominance. What this basically means is that whenever a dominant is with a recessive, it will hide the recessive. So whenever a big letter is combined with a little letter, you're always going to be able to see the big letter phenotype ahead of the little letter. So every gene has 2 or more alleles. Alleles are what Mendel thought to be factors. He called them factors. He called genes factors. He really didn't know at the time, but he had a very good understanding. that these characteristics combine to give us our phenotype. All right? So there is one gene for seed shape with 2 alleles, one allele for smooth, the other allele for wrinkled. All right, this illustration just shows us an example of height for pea plants. So we have the parent generation where we have the organism being pure tall and then the other organism being pure short. When you combine them, look at the F1. Well, you wouldn't think that there was any short traits at all in that first generation, but guess what? There are. They just don't appear. When do they appear? They appear in the F2 generation. Okay, the possibility of them coming up doesn't have to always happen, but the possibility of them showing up again is going to appear in the second generation. All right, guys, the principle of segregation in your book is written as the law of segregation. They both mean the same thing. Okay, some resources will say the principle, other resources will say the law of segregation. Remember, that word define means to separate. So what's separating is traits from mom and traits from dad are separating. Okay, we all have traits from mom and traits from dad. So during this process called meiosis, which we're going to learn about later on, mom's traits and dad's traits separate from one another and they go into sperm and egg cells, which are then going to combine with either sperm or egg cell to make a new individual. Principle of independent assortment can also be written as the law of independent assortment because that's how it's described in your textbook. So what this again means is that different traits like height, for example, and maybe eye color don't have to go into the same places. Okay? They assort independent of one another. The only time different genes go to the same area or the same place is when they are closely on chromosomes. Okay? Otherwise, one dominant trait does not have to always go with another dominant trait. Height does not have to go with hair color, for example. They assort independently. All right, so all this diagram here exhibits is, if you look here, this cell, then it distributes into this one, and by the end of this process, we have 4 different cells. Now, these cells have the same kind of genes in them, but this cell has a different combination. Probability plays a very important role because we're all talking about the chance of different traits assorting or separating or segregating themselves into new organisms. Okay, so again, the Punnett square created by R.J. Punnett is the name of the person that started using them first to figure out probability and potential of what traits would go into organisms. Okay, here are some of the main or major vocabulary that we're going to be introduced to in this chapter. And it's very important that you guys differentiate between homozygous and heterozygous because a lot of folks are unable to do so. And also genotype and phenotype. That is also another important distinction that we need to make as we're going through this chapter. The reason why we have the letter B in parentheses, okay, or quotations, is because we can supplement any letter in there. We can use an M, an N, an O. We can use A's, okay? That's why it's, in the quotation marks there. This slide is a little bit jacked up, but here is what it is asking you to do. It is asking you to do a Punnett square cross between 2 heterozygous parents. Capital A, lowercase a, capital A, lowercase a. And it's asking you to figure out the different genotype pairings that occur within the Punnett square. So let's try it right now, please. So what we have here is a 3/4 of a possibility that we're going to get dominant characteristics and a 1/4 of a possibility that we're going to get recessive characteristics, or it's 3 to 1. Now, that is the phenotype ratio. the genotype ratio is 1 to 2 to 1. What does that mean? It means you have 1 homozygous dominant, 2 heterozygous dominant, 1 homozygous recessive. Now, if you add those up, they equal the 4 possibilities you see with that monohybrid Punnett square. So we've already gone over how to do dihybrid crosses. We went over this all this week, so you guys have a very good idea of how to set it up. The 2 different methods that we used were the FOIL method and the Punnett square method in determining the possible gametes. Remember, each parent contributes 4 possible gametes when we're doing dihybrid crosses. So if we look at a dihybrid cross between heterozygous parents, we know that we're going to get the ratio for phenotype 9, 3, 3, 1. All right, so the product rule is different from what we've gone over. If you use the product rule, you don't have to do a dihybrid cross. So in certain instances, when they're trying to ask you to figure out the probability of certain things and they don't say do a dihybrid cross, then you can use this product rule. Let's look. For example, if we want to know what's the probability of having red and tall, okay, then all we need to do is take 3/4 red, multiply it by 3/4 tall. That gives us what? 9/16, right? What if we want to know the probability of having red and short? Then we take 3/4 from red, multiply it by 1/4 for short, and what do we get? We get 3/16. Okay, what if we wanted white and tall? We take 1/4 for the probability of being white colored. We take 3/4 for the probability of being tall. We multiply them together, and what do we get? 3/16. Lastly, if we want to know the probability of having white and short, 1/4 for white times 1/4 for short, and that gives us what? 1/16. Okay, so the product rule is a quick way of doing the math that you need to do to get the probabilities instead of doing a Punnett square. Okay, so here we show the combinations that I was just talking about. Okay, so again, say that we were doing a trihybrid cross, which does exist. And in more advanced classes like AP Biology, we might have to do a trihybrid cross. But you guys could actually do a trihybrid cross. The only thing you would need to do is do 3 independent Punnett squares and take the percentages and multiply them times one another, and that would give you the probability instead of doing a 64-box Punnett square. So, method 2, we've done this method already. This represents the FOIL method. Okay, this what you see on the board and you just took a picture of represents the FOIL method. First, outer, inner, last. And that's how they got the 4 possible gametes that you see there in red on the bottom. From that, we know what goes on the outside and what goes on the top for our 16-box dihybrid cross. And if we do the probabilities, we count how many of each, we know that we are always going to get the phenotype of 9, 3, 3, 1 whenever we're dealing with parents that are heterozygous for the 2 traits. All right, so it says beyond basic crosses. Now these are more complex things that happen in higher organisms and in some cases some plants too. Now say that Mendel did his experimentations on any kind of animal, or he used snapdragons for example. Well, he would have probably gotten a little confused because there are different things that happen in higher order organisms. It's not just simply dominance over recessiveness. So we have these special cases, or in some instances they're called alternate patterns of inheritance. Okay, with incomplete dominance. Now this is different from basic dominance, okay, that we learned about in the first part of the chapter. In incomplete dominance, the dominant trait is not completely dominant over the recessive. So red is the dominant characteristic, white is the recessive characteristic, However, when red and white are combined, you get a color like pink. Okay, when that happens in organisms like the snapdragons, then it is called incomplete dominance. Okay, again, different from the pea plant color, where you had purple being dominant over white. It was a clear-cut thing. This is not clear-cut. That's why it's considered a special case. Is codominance. Now, in codominance, both characteristics display themselves simultaneously. Okay, in the case of this cattle, this same thing happens in some horses as well, where you get a color called roan. And that is because you have both the red coloration and the white coloration both appearing at the same time, and it appears like a orangish color, and they call that roan. And in certain flowers, you see the purple and the white, and it's a speckled looking color. Now, to our eye, it looks speckled, but both of those colors are displayed simultaneously. Blood type is also an example of two things: codominance, and the next thing we're going to learn about is multiple alleles. All right, today we're going to be doing a couple of things. We're going to finish up our notes, and then we're going to go into discussing blood type. But the thing about blood type is that it is both codominance and multiple alleles. What that means is that there are three different alleles or three different traits that make up the copy of the gene for blood type. So although you're only going to carry two copies of the gene, there are three things that make that gene up. And it shows you here how to write blood type A and B. For example, if I'm writing blood type A, I'm going to write a capital letter, and then I'm going to either put a capital A or a capital B superscripted. Okay, that means above it to the right. If I am writing blood type O, I am using a lowercase I. Now, the reason they do that is to distinguish it between the other types of genetic studies, and also because it has something to do with the immune system, immunology, etc. Okay, so we're going to get some practice on that, but it shows you on the board there how you're supposed to write the alleles when we're dealing with blood type. Okay, when we have a situation where there are many different genes that control a trait's expression, then it's called polygenic inheritance. So things like height, eye color, skin color, they aren't attributed to only two alleles. They're attributed to several alleles all working together to determine what is viewed as complex in those phenotypes. Okay, so this is just a diagram showing us, we see the variation in eye colors here, and we also see a chart. This would be a 64 box Punnett square. Okay, and it's showing you the various variations that the sperm cell can have with allele combinations, and then the variations for egg cell. So in order to do this, we could do the product rule. That would be an easy way of determining it. We wouldn't do a 64 box Punnett square in this class, but it shows you how all kinds of variations can exist when you're talking about skin color. Because each individual gives up to three different alleles for the traits. All right, some environmental factors also affect how certain traits are going to express themselves. So, or in other words, phenotype does have an environmental factor. Okay, so in this instance, temperature, and the temperature in the various parts of these animals' bodies determine whether or not the colorations are going to be expressed. Okay. All right, so this illustration will give us an example of how temperature and environmental factor will determine coloration in certain organisms. So this is probably like an ice pack. And what they did is they put the ice pack, not sure how much time they held it there for, but they put an ice pack on the rabbit there, and what they noticed is that depending on the temperature of the ice pack, coloration, that black coloration would appear on the rabbit. Okay, so just showing you again that environment does affect and factor in to how phenotypes might develop in animals and other organisms.